cool. I'm so glad that we could like all fit in this room and all be together. It's like a big room, but it is what it is. Okay, so we've already covered a lot of material. It's like kind of crazy because I met with like half of you yesterday for lab, and I'll meet with the other half of you for lab today. The lab's gonna be really short, like 15 minutes, just to go over the instructions. We'll do that as you go. All right, that's interesting. So, yes.
Can you take that white one and like hold it down and turn it off for 30 minutes?
Cool.
You don't like hold it, yeah.
Thank you. All good. All right. Interesting things, new room. All good. So we've already covered like a lot of material with vision, I know. Are there any questions about what we've covered with dark adaptation, light adaptation? Just spot like over here, or over there. Okay. All right. No questions? You guys are good? I mean, I feel like that was a lot of stuff that I threw at you last week, so good. Okay. So I'm doing this course a little bit differently than I've done it in the past. So in the past, it was a Monday, Wednesday, Friday course, and now we're doing Tuesday, Thursday. Unfortunately, this is the only time I could get this room that we could all be together, so I'm sorry that it's 8 o'clock in the morning. I know it's early, so I apologize about that, but I think it's a good thing for us. I've reorganized the course since we were switching, since I had to kind of mix all the lectures up. And so we're doing it more like the textbook flows, starting with vision, because normally I start with touch, and we do that first, and then we do the chemical synthesis. It's kind of going to be in different order. It's kind of new, which is kind of exciting. It's kind of fresh and new stuff. So vision, we talked about transduction. We've talked about the retina, processing in the retina, lateral inhibition, right? Yeah, blank looks, good looks, head nods, good. Okay. Center surround receptive fields. Okay. All right, so today we're going to start to move out of the eye, because like we haven't even seen anything yet, right? We're just in the retina. We haven't even gotten to the brain yet. So today we're going to move to the LGN and hopefully get into the cortex and look at how this stuff is actually being perceived here. All right, so what are Mach bands? Anyone know what Mach bands are? It's a tiny little thing in your textbook, but I think it's really cool, so I want to talk about it for a second. So if we look here, we go from dark to light. And there's actually a gradual transition from this darkness intensity to this brighter intensity. And so it really looks like, if we were looking in the retina, what's shining on the retina, it's a little bit of review, right? If we're looking at retina, what's shining on the retina, we're starting at 2s, and we're going to go to 6s, and in the transition, we're just going to go 3, 4, 5, 6, right? Straight, linear sort of projection. But what happens at points D and B? What do you see at where it's marked D and B? What do you see where it's marked B? What, Nora?
A line.
A line, it's brighter, right? You see like a brighter line right there. And what about where it's at D?
You see a darker line.
A darker line there, right? What's causing that? These are called Mach bands. What's causing the Mach bands?
I forget specifically what causes it, but I know it's basically your brain trying to like delineate between the two areas.
Yeah, so we talked about this I talked to a camera last time about this, right? So when we talk about lateral inhibition, what's the point of lateral inhibition?
So you can
Jen? To see edges better. So what's happening at D and B? We have an edge, right? We're starting to transition, even though the actual light intensity doesn't show that, right? Because of lateral inhibition and center surround receptive fields, right? We end up having a perception where at this edge, it gets darker because there's less inhibition from this side than this side, right? Which makes it a smaller signal going to the brain. And over here, because it's brighter, there's more inhibition. More inhibition means that it becomes lighter, right? And so we get that bump. So we get that edge perception that happens in both places, right? All right, so that's just a little review of what we did last time in terms of lateral inhibition and center surround receptive fields. If you need to go back and rewatch that lecture, do it, because that's a really important concept in terms of the fact that the signal that goes to your brain, and I mean, I can't stress this enough, but the signal that goes to your brain is not what you're actually seeing. Like, that doesn't exist. It's not actually lighter right here. But we perceive that stripe there because our retina, our eye, changes the signal before it ever even gets to our brain, which I think is a pretty cool thing. All right. So we have two images that come in through our eyes. Because we have two eyes, we have binocular vision. And those two images, if we have something right here, it's in our fovea, our center of vision, and it's striking each eye, we have to take that representation of that image and consolidate it into one thing, right? Into one thing that's going to get to the brain. So we have to take half of each eye and move it to the other side of the brain because we want to take everything in our right visual world and put it on the left visual cortex, everything in our left visual world and put it on our right visual cortex. Okay? It's another sort of just image of that pathway where we're taking and sending that signal then through the optic chiasm is where we're going to cross over, consolidate those images, and then the next stop for that signal is going to be the LGN, the lateral geniculate nucleus of the thalamus. All sensory systems are going to go through the thalamus. What's the job of the thalamus? Who remembers that from biopsych?
Kind of regulate your senses. I forgot the word. To filter. Yes.
To filter. Thank you, Beth. Appreciate it. So, yeah, to filter things, right? So things that are not as important, we're going to filter it out so our brain doesn't have to worry about that. So all our sensory systems are going to go through the thalamus. I put that bottom-up signaling worksheet on the website. And I think that would be really good for you to fill for every sensory system, right? You need to know, you maybe make a table that says areas of the thalamus. You need to know what area of the thalamus all the sensory systems go through, right? So LGN is vision. Okay, so I like this image. I've shown you a bunch of different images from the different textbooks. I like this one because it shows you like 1 through 9, left visual field, right visual field, how that gets represented 1 through 9, inverted, right, through the lens on each eye. And then it shows you how those signals then get consolidated through the optic chiasm so that we get our entire right visual field on the left visual cortex and our left visual field in the right visual cortex. All right. There are some secondary pathways. There's 3 secondary pathways that are not involved with our primary vision, but go to different places in the brain. The first is the superior, well, not first, second, third, but one of them is the superior colliculus. Does anyone know what the superior colliculus does? Yes, Jalen. Yes, it helps us with visual tracking. Sorry, I forgot to turn my phone off. All right. It helps with visual tracking, right? So we're sending unperceived, subconscious, when we talk about subconscious, it's not going to our cortex. We're not perceiving these signals to help us focus on things that are moving in our world, to direct our eyes to put things that are moving into our fovea, right? There's also an area of the midbrain called the pretectum that's receiving input that helps us control how dilated our pupils are. In bright light, our pupils become constricted. In dim light, our pupils become dilated. That's not a conscious perception. There's a direct feedback of how much light's coming in to sort of control that muscle. So it's reflexive. And then the hypothalamus. Why do we send signals to the hypothalamus? What's that about? What all is the hypothalamus involved with?
Revision of the sleep-wake cycle.
The sleep-wake cycle, yes. So we're sending signals directly to the suprachiasmatic nucleus, and so bright light in the morning helps us reset our sleep-wake cycle. All right. And then finally, I just want to note that at the optic chiasm, that's sort of the neuroanatomical delineation between peripheral and central nervous system. So somebody 100 years ago made a decision that that point is where we're going to call, now it's in the central nervous system. So we call this tract here the optic nerve. Once it gets to, we don't have nerves in the central nervous system, we have tracts or pathways. So now it's called the optic tract on this side of the optic chiasm. So it's kind of a weird thing. I just wanted to point that out so you understood that. All right, so we talked about the hypothalamus as circadian rhythms. How about the superior colliculus for automatic eye movements? And then in the pretectum, this area is called the Enger-Westphal nucleus. And again, that controls the, through cranial nerve 3, the oculomotor nerve, controls how constricted your pupils are. Right? And this little structure here is the cerebral aqueduct, right? So CSF flows right here, and it's very, very close to this nucleus. And so when you have inhibitory types of drugs in your body, like alcohol, it seeps out of the cerebral spinal fluid and this right here and affects your ability to constrict your pupils, which is why if you get pulled over or suspected for driving under the influence, the police officer is going to shine a light in your eye and see if it goes from being dilated to constricted. If you don't get that reflex, if that reflex is inhibited, that's an indicator that there's inhibitory drugs that are negatively affecting that synapse there. Right? So that's the Enger-Westphal nucleus. Okay. All right, so back to our visual pathway, our primary visual pathway. So half of each retina is going to go to, is going to cross over and go ipsilateral to, or contralateral to the other visual cortex, right? And it's important to know that this retinotopic, what we call retinotopic, so based on where you are in the retina, fovea, we can go like 10 degrees, 20 degrees, 30, further around the whole retina, that organization of where things are in space, because that's really what this is, right? Things that are at 1 are coming from way over here in my visual field. Things that are at 5 are in my fovea, in my center of vision. That retinotopic organization is maintained in the LGN and in the striate cortex. So we're maintaining where things are in space. That's the way our visual system is organized so that we can identify where things are in space. All right, so I mentioned striate cortex. Yes, Jim? Yes, so it's, so here is the medial, right, or nasal, next to the nose, half of each eye is what's receiving that signal that needs to go contralateral, that needs to cross over through the optic chiasm. And the medial half, from the fovea to the medial part, stays ipsilateral. Okay? So when we have, say, something at point 8, it's coming in and activating point 8 here, but it's also activating point 8 on the nasal side here. And we have to take those two images and consolidate them, right? If it didn't get consolidated, what would happen? What would happen if we didn't consolidate those images? You'd see two things, right? And you could do that, like if you gently like push your eyeball out of alignment, like down here, what happens? Right? You can see two things. Anyway, don't do that. Okay, I have a better demonstration. So take your finger and put it about 6 inches away from your nose. Take your other finger and put it as far away as you can get. Now, focus on your close finger. How many of the other finger do you see if you focus on your close finger? Focus on your far away finger. How many of your close finger do you see? So we'll talk about this after the first exam. In the second section of the class, we'll talk about depth perception, and we'll talk about corresponding retinal points. These are called corresponding retinal points. So this 8 corresponds with this 8. When we focus our depth perception on non-corresponding retinal points, it creates diplopia, double vision, and you see two of an image, which is what we were doing right there. Cool? I don't know, I think this stuff is so cool. I get a little excited. I'm sorry it's 8 o'clock in the morning, but I get a little excited about it. It's like amazing that our brain does all this stuff. It's so cool. All right, so does anyone remember how many neurons are in the optic nerve? We talked about having 120 million receptors. 120 million receptors in the retina. How many neurons are there going to the brain? You're like, man, I didn't know he was going to ask me stuff from that video that I didn't watch. Any guesses? More or less than 120 million? Less, right? Because we have the center surround receptive fields. What's the purpose of center surround receptive fields? Why do we have center surround receptive fields? Because it makes things more complicated to learn. Main reason.
So you can do more with fewer. Each one has 2 settings, so you can do more with fewer.
Yeah, because we have to take 120 million receptors, converge them into 1 million neurons in the optic nerve going to the brain, right? And so center surround receptive fields allow us to converge the signal, right, without losing everything. If we took say we take 120 receptors and put them into 1 neuron, and we don't have center surround with the amacrine cells, then we can't tell where light is in that receptive field of 120 receptors. But if we have center surround with amacrine cells, we can tell if it's in the middle or the periphery. We get 2 signals, and we have some better clarity of where that signal is, right? So we converge, and this is going to be a big, thing that we talk about, right? We have receptors. We have to converge them into 1 million optic nerve. But when we have a little bit of divergence at the LGN. But when we get to the visual cortex, to the brain, we can now do this and have divergence, because now we have 250 million V1 cells, right? So we can now, that we're in the striate cortex, we can open the system back up, and these 1 million neurons, it's actually, at the LGN it goes to 1.5 million. So there's a little tiny bit of divergence at the level of the lateral geniculate nucleus, so that signal gets a little bit bigger. It's not like 2 pathways, it's like 1 pathway But then when we get to V1, we can send signals to lots of different places and diverge the signal and increase our processing power to be able to have that visual acuity so that we can see really fine details. V1 we'll talk about today is involved with, so basically you can kind of think about your brain kind of like a computer. I don't know, how many of you have ever taken a computer science class? Raise the hands. Great, not many. Okay, so does anyone know how computers deal with information? It's binary. What does that mean? 1 or a 0. They deal with like on-off. It's electricity, right, in a computer. 1s and 0s. It's called binary computing. Man, y'all should all take a computer science class. Okay, 1s and 0s. Well, that's like our brain, except we have like 1s, 0s, and 2s because we can have excitation, baseline, and inhibition. We actually get 3 signals, right, instead of just 1s and 0s. But we have to take what we call an analog world, right, and basically convert it into action potentials, go into the brain, which is kind of digital. It's binary, kind of, right? On-off signals, center surround signals. And then when we get to the V1, we have to reassemble this analog world that's so rich with color and objects and texture and everything. We have to reassemble that in our perception. And that's the job of V1. So V1 has to sort of rebuild from those digital signals what we're seeing out there. Right? And so we're going to do that through primary feature detection, things like angles of lines, movements in certain directions, lengths of lines, and then we're going to build those into more complex shapes, right? So I'm going to take your face and break it down into lines and then reassemble it into eyes, hair, forehead, mask, the whole thing, right? All right. And that's what extra striate cortex is doing. So we have extra striate cortex right here. Does this sound really complicated? A really complicated process to do in your brain? It is. I mean, look how much of your brain, basically almost a third of your brain, is devoted to vision processing, right? When we think about the brain, we think about thought, personality, emotion, judgment. All that's happening like right here in this little area. And we have a third of the brain back here that's trying to figure out what are we seeing and do it in real time so there's no lag, right? There's no lag. It's not like something that we saw happen seconds ago. It's doing all this really fast processing. So huge amount of brain processing power is dedicated to vision and understanding vision. All right, balance, back to the balance. First place, so we get to the balance, this is the first place that we have top-down influences on our perceptions. What's a top-down influence? Yes, Suzanne?
It's like what you perceive it to be down to like the actual physical.
Yeah, so it's the way our brain influences the bottom-up signal. So when we talk about bottom-up signaling, we're talking about the digital input, basically, that I was talking about, right? Those action potentials coming in. But those action potentials get shaped and changed based on our experience, based on our memory, right? Did my little demonstration work on the video with the mouse and the man's head? Did y'all see that? Did you do it? Did you play along with me? Okay, thank you. All right, that's an example of top-down influence. And the LGN is the first place the brain starts shaping those signals. And it shapes it on attention. It shapes it on whether or not you're paying attention. Have you ever been like tired driving home from college end of the semester and you're like, man, I really just don't remember that last couple of miles on the highway. That's kind of scary, right? Because your brain like literally filtered out that information. All that visual information as you're rocketing down the highway, right? And we've all sort of had that experience, right? Where levels of arousal can affect what's coming into our brain. And this starts at the LGN. So again, we have 1 million neurons coming into the LGN. We have 1.5 million, so we start to get additional signals added in coming out of the LGN, right? And the brainstem, that locus coeruleus for vigilance and arousal, is bringing in excitatory and inhibitory signals that are sort of regulating what information gets to V1, gets to perception. Right? All right. The cortex also comes down here and supplies information. So we have two things that can filter signals getting to our perception. Our overall arousal level, that's brainstem, lower brain functioning, okay? And the cortex, which is selective attention. If you're not paying attention to something, you won't see it. And again, we'll talk about that later when we get to sort of upper level processing. All right, so here's one LGN, a stained picture of it. And what you can see, so we have these ganglion center surround receptive fields like donuts, right? We have a center that's either on or off and a surround that's the opposite. So I'll just talk about the center being on and the surround being off, but you know it could be the other way because everything's complicated in the visual system, all right? Those donuts are center surround receptive fields are preserved. They're still there in the LGN. And we still have that retinotopic organization. So point A, B, C across our visual field on our retina is maintained as A, B, C across here in our ganglion, in our LGN. What's different in the LGN is we now have input from both eyes coming in. And we have to consolidate A, this point A, that's hitting our left and our right eye, has to get consolidated. So what we end up having are layers of contralateral and ipsilateral input. And there's actually 6 layers, and it does a weird thing here. It goes from ipsilateral, and then this layer's ipsilateral, and then contralateral. It kind of flips there. That's actually important because there's 2 sort of sections of the LGN. Whoops, let me pull all that up. So in layers 1, 4, and 6, that's not right, 1, 3, and 6. I'll fix that. We have the contralateral, and then layers 2, 4, and 5, we have the ipsilateral, right? Those numbers should be switched. I'll fix it. And that's because this area that is in the first 4 layers is called the parvocellular area, and primarily receives input from our cones and our fovea. And the magnocellular layer, where it switches, where we have ipsilateral and then ipsilateral, is receiving primarily input from our rods and our peripheral cones. So the main thing that we care about in the visual system is information right in front of us, right? That's where our detail is. So if I'm looking straight ahead, I can see Zach, but I really can't see his face. I see a body over there, right? If you look directly at me, you probably can't read the words on the screen. You can see there's a screen there, but you can't read the words on it, right? Because it's not in your center of vision. So we devote a lot of brain and general processing information to our fovea, to our center of vision, and not as much to the magnocellular peripheral vision. All right. There's also these things called K cells that we don't really understand what they do, which to me is remarkable. The most amount of research in neuroscience has been, in the sensory systems, has been devoted to understanding how vision works. It is the most studied system, and we understand it down to lateral inhibition with horizontal cells and amacrine cells and all this stuff. But there's still parts of it that we don't understand yet, which to me is, again, one of these amazing things about neuroscience is that you have these huge, vast books of information, of knowledge that exist, yet it's almost like we've only scratched the surface. And there's things that we know exist that we don't know what they do. And then I know that there's probably more things that exist that we haven't discovered yet. We haven't even identified or found, right? Which is just wild to me. All right. So P cells are center of vision. They tend to have a slow, steady response. We're trying to get details from them. They deal with color. M cells in the periphery tend to have more like on-off, fast, abrupt signals. They're more interested in motion. We want to see if something's coming, moving towards our fovea, right? We want that signal to go to our superior colliculus from our magnocellular layer to put our eyes onto the, Marianne, can you do me a favor? Thank you. To put our eyes on the fovea, right? To put that thing that's moving in our periphery into the fovea. All right. So, lots of different pictures from different editions of your book. They just came out with a new edition of the book. If you have an old edition, you have a new edition, it really doesn't matter. I like these textbook manufacturers, all they do is they like create a new edition and they change a figure or two. They just update what it looks like, and then they charge you a lot more money for the new book. It's just really a ripoff, and I'm sorry that y'all have to like deal with that. But, so I don't care what edition you have, the information is all basically the same. Some of the figures are going to be different, but they're all saying the same thing, right? We have M cells that are coming in and they're going to areas of motion. We have P cells that are coming in, they're going to areas of detail and identification. I like this figure, which is from a really old textbook, because it's simple. I mean, these are like, I don't know, cluttered with a lot of stuff and a lot of things you don't really need to know, right? So I like this one. And basically it shows you that you have a parvocellular pathway and magnocellular pathway. Both go to the LGN. The magnocellular splits off, sends some movement signals to the superior colliculus. So if something's moving in our periphery, we can focus our eyes and get it in our parvocellular pathway. We get combination of the visual fields here. We still have separation of parvocellular and magnocellular though. And we have our detail and our sort of movement. If we want to simplify what are those things doing, you can just basically go center vision and movement, peripheral vision. Filtering of the signal there. And then we get to V1. We get to V1 and we can now diverge our signal to those 250 million neurons. And then from there, we reconstruct what we're seeing into objects. And then once we have an object rebuilt, we want to identify what is that object, right? Who does that face belong to? It's Lexi. Okay, I can like see the eyes, put it together, I know it's different than Daisy. That's what's going on in our association cortex and our ventral pathway there. All right, so as I mentioned, V1, primary cortex, it's the occipital pole, the very back part of the occipital lobe. And from there, the signal rebuilds from primary features into complex objects and movement and color, and things get associated. And we have two pathways that come out of V1. And we have different areas that are dedicated to particular complex processing, like creating objects, associating colors with those objects, movement processing in V5. And then we have a pathway that goes ventral into the temporal lobe. What is the temporal lobe known for?
Hearing.
Hearing, yes. But in vision, what is it? Or related to vision? You're correct. Hearing is there. We'll get to that later in this course. What's deep inside the temporal cortex? What structure? This site was so long ago.
Is it the hippocampus?
The hippocampus, and what is the hippocampus known for?
Memory.
Memory. And so what's found in the temporal lobe? Memories, those neural networks, right? And so vision, what do you think vision's doing in the going ventral into the temporal lobe? Yes, that's what a memory is, right? You know, what is this thing? If this thing is shining on my retina, how do I identify it? I reconstruct it into an object, and then I send it down to my temporal lobe and access the memory that this is a stapler. And along with that, I know what sound it makes when you click it. I know how to use it, right? And then we also have a pathway that's going up towards the frontal lobe. What's the frontal lobe involved with? Higher cognitive function, right? Decision making, motor system. This is, what do I do with that thing? If I recognize that as a stapler, and I'm like, oh, I have some papers I need to staple, I can now access through this dorsal pathway how to use that. What do I do with it, right? So we have these two pathways, this pathway that's coming up towards what do I do with it, and a pathway that's going down into what is it. And those things are happening simultaneously, and they're talking to each other. They're talking to each other so that the temporal lobe goes, that's a stapler, and the frontal lobe goes, oh, I know how to use a stapler. Right? And you can have like a really weird looking stapler that you have to figure out it's a stapler, and then you've created a new memory, but you still didn't know how to use it. All right. So this is where we get a little bit complicated. We have to go from these donut center surround receptive fields and deal with this divergence. Okay? So the whole point of having center surround receptive fields is if I have a point of light that comes in, so here's this center surround receptive field, and they overlap with each other, and it activates the center, right, I get a positive signal. And if I have a point of light that's a little bit over here, and it comes in and activates inhibitory, I get inhibitory. Here, because they're overlapping, I'm getting two inhibitory signals, so I know exactly that it's on this side of the center surround and not that side, right? So we're able to take these on-off, digital on-off signals, and process them with all those big neurons, the 250 million neurons, to reconstruct our world. All right, so we have all these center surround receptive fields in here, and we have so many of them overlapping that we start to create areas of excitation. Like you can see this excitation that's sort of surrounded by inhibition. And so what we end up having in the visual cortex, because we can diverge, because we can take these 1 million outputs and send them to lots of places, right? We're going from 1 to 250, big expansion, that we can now have a single neuron in V1 that's receiving all of these excitatory and all of these inhibitory, and its receptive field now looks like a vertical line. It's not a donut anymore. It's become an orientation of a line, in this case a vertical line. Do you all understand how that happens? It's a really important concept. How we go from donuts to lines. All right, so Hugo and Weasel, really important names. These are names you'll see on the GRE. You'll see it on your psych assessment when we do the major field test for our senior assessment. So I don't really care about making you memorize famous people, but when there are people that I know you're going to see on tests, I'm going to point them out so that you know, hey, if I see Hugo and Weasel, I'm looking for anything to do with vision, anything to do with receptive fields. And that's the answer on the multiple choice test, okay? Which is why that's important. They won the Nobel Prize for this work. In 1981, they were awarded the Nobel Prize in Physiology and Medicine. So they were starting with a map to take electrodes, put electrodes in a cat's LGN, because we have to have the right research model to study what it is we want to study. Rats have really poor vision. Not a good model to study vision, right? They have pretty good taste. They're a pretty good model for taste and smell, but not a good model for vision. And so cats have really good vision. And so they were a really good animal model. So they're putting electrodes in a cat's thalamus and then putting points of light, and they were saying, okay, this is in that neuron's receptive field. If I move it far enough away, it goes out of the receptive field. I can map this donut, like inhibitory area and center excitatory area, and map those receptive fields, right? So the LGN was responding to these spots of light in these center surround receptive fields. Then they went to the cortex, they went to V1 and started to map V1 and try and find cells, receptive fields. And these spots of light that they were shining, so it was in the 1950s with a slide projector, right, weren't causing responses in V1. But then they noticed that when they would put a new slide in that had a point of light at a different place, sometimes the cell would fire, and it would fire as the slide was sliding down, and it turns out that they identified that it was actually the edge of the slide, that the cells in V1 weren't donut center surround. They responded best to lines of different orientations. And so when they switched their stimuli from spots of light to lines, they could now map these receptive fields in V1. And that's what they got the Nobel Prize for. They were the first people to understand that the donut center surround receptive fields in our periphery, in the LGN, in the thalamus, diverge into angles of lines receptive fields in V1. And so what's important is where. So we still have where. We have a receptive field. If that angle line's here, if I move it over here, it's out of that one neuron's receptive field and into some other neuron's receptive field. So we maintain the retinotopic mapping of our visual world, right? But now we have angles of lines. And it wasn't just angles of lines, but also movement. We'll talk about that as well. So if I have my electrode in V1 in a neuron that has its receptive field in that vertical orientation, that neuron's going to fire the most when I put a vertical line there. But as I rotate that, now I'm in the excitatory, but I'm also activating inhibitory, and the signal goes down. And if I make it totally the opposite of what the receptive field is, a horizontal line, I actually get more inhibition than excitation, and so I actually can turn that neuron off and inhibit it. Okay. So this then allows us to create what we call tuning curves. There we go. Tuning curves, so that here, this neuron that we were just talking about fires the most for a vertical line. And as we make that line less vertical, as we start to rotate the angle of the line all the way to horizontal, it fires less. So it fires the most for its orientation that it's tuned for, and then it fires less. We're only talking about one neuron that's firing for a vertical line. Now, if I pick my electrode up and I go over just a tiny little bit and go back down, I'm going to find a receptive field that's now tuned to this. It's going to fire the most to this angle of line and have a tuning curve that's shifted over. And so we do that across what we call a hypercolumn for every possible orientation of line at that particular place in space. Which is why we need 250 million neurons here. Because we're doing this for every possible orientation of line for that particular place in space. And then if I pick up and go over again, I find a new receptive field, so it's no longer right here, it's now a vertical line that's over here. And now I get another hypercolumn with all possible angles of lines at this receptive field, retinotopic. All right. tuning curve. So, just to make this clear, for this particular place on the screen, if I were to show that line here, I would be out of that, like that neuron's receptive field, and I'd have to go search and find a neuron that has this receptive field. But for this place in space, that neuron is firing the most for a vertical line. As we move away from a vertical line, it goes down, right, even more, until we get to where it's really not responsive at all. If I'm going perpendicular to the cortex with my electrode, all the cells coming down here are going to fire and have that orientation curve, in this case for a horizontal line, the most, right? And like I said, if I pick up and come over here, I'm going to find cells that respond to a slightly altered horizontal line. And as I go across, I can have every possible combination. And if we were to stick an electrode in and go across these columns, so this is a column. If we were to go across these columns, we start to see, okay, this is this angle of line, then it changes to this angle, then to this angle, then to this angle. So you can go across and see, measure these changes in the receptive field. Good? All right. So here is what I call the hypercolumn, where we have, again, left eye, right eye. So this is the same place in space that's hitting both my left eye and my right eye. They're matched up, so I have single vision, not double vision. And for that particular place out in space, I have a column that's going to respond to every possible orientation of line. This is sort of another example of trying to show that, where if I have a blue and a green dot, they're coming in and shining on each eye. Here's where the green dot receptive field is. Right next to it is the receptive field a little bit over for the blue. And we have two hypercolumns from the left and right eye, left, right eye, that are being consolidated, and any angle of line within that little circle has a column of cells that will respond to that particular angle. So I can know if it's a vertical or horizontal or whatever angle of line there is. Good? All right. So this collection of possible columns of angles of lines is called a hypercolumn. A hypercolumn. And if we look at, we can put a tracer in one eye and to stain neurons, and not put the tracer in the other eye, and then we go look at V1. In V1, you can actually see like left eye, right eye, left eye, right eye, left eye, right eye, left eye, right eye. It's striated, striped. We call it striated or striped because it has these stripes of left eye, right eye, left eye, right eye that are consolidating those images there. All right. We have these overlapping receptive fields, right? This looks complicated. That's only 25 ganglion cells, right? We have 1 million ganglion cells, right? So it's lots of overlap. We can generate any possible angle within there of lines. So if this is let's say this is the hypercolumn for this vertical line here. That means that all of the on, we're just looking at, so now we're not looking at on-off, we're just looking at the on parts. We're not going to worry about the off parts, we're just going to look at on. So a line that's right here is going to stimulate all 5 of those on cells, and all 5 of those on cells are going to converge on this column. Right? This is why we get this divergence to 250 million, because this one green line, if we have a slightly rotated one like this, which would go to this column, right? We move over a little bit, we go from vertical to slightly rotated. The green is still part of that line, right? The green, the center of this receptive field is going to be part of any line. A horizontal line, did I get to a horizontal line? I don't get to a horizontal line. But any line in here, so the green neuron is going to send axons to every possible orientation because it's a center, and it will they'll be stimulated by any possible rotation of that line. The red cell, right, is only going to the vertical because as soon as we move out of the vertical, the red cell is no longer contributing to any of these receptive fields. Does that make sense? Okay. It's really complicated. I'm trying to figure out a way to explain this divergence. And so here we have a line, and this cell is now part of this orientation, but this cell is also part of this vertical in a different receptive field though. So there's another hypercolumn that this neuron is projecting to, and it's contributing to vertical types of lines for this area. So it's not just divergence for this one hypercolumn, but it's other hypercolumns, other receptive fields that those neurons can be part of. Really complicated. It's like amazing that our visual system develops like this. Like how does it know to send these axons? I mean, it's just, it blows my mind. I can't fathom like how our brain develops and sets this up. But it's really cool how it does it. And it's really cool that it allows us to go from these donut center surround receptive fields to being able to see lines out there in space. All right. So far, all we've talked about are simple cells, angles of lines. That's called a simple cell. You have a tuning curve, and it just matters what the angle of line is within that receptive field. Because we have that divergence to 250 million neurons, we're able to create these hypercolumns in V1. Okay, good. We haven't even talked about color. So this is how complicated it gets. Is that color is sort of laid on top of these hypercolumns. And color is sort of weird. We talk about it in terms of blobs because it's not like very orderly. Like these are green, a hypercolumn that's spun into green angles of lines, and then over here it's red, and over here it's blue, and down here it's yellow. It's just kind of like blobs. It's not very orderly. We have this very orderly retinotopic thing, but color is complicated, right? And I don't know that we really understand how our brain's able to process all that. All right. Simple cells, angles of lines, that's it. Complex cells can detect two features. It detects the angle of line, but the receptive field may be bigger. So it's an angle of line that's within an area. So now we're able to sort of detect where that angle of line is. And for that bigger area, we're detecting if that angle of line moves in that area. So now we have, it's orientated only for a horizontal line. It fires. It won't fire for a vertical line. Okay? And it's only oriented for a horizontal line that's moving down. And if it moves up, if it's a horizontal line but moving in the opposite direction, it won't fire. So now we're coding for two features, movement and orientation of line. Here we see another receptive field of a complex cell that is responding only when it's this angle, doesn't respond to any other angle, and only when that angle moves in an upward fashion. If we move it in a downward fashion, it doesn't respond. Good? Simple cells, static receptive field, angle of line. Complex cells, angle of line, but now direction-specific movement. Hypercomplex or end-stopped, you'll see both of those terms, end-stopped or hypercomplex, now have 3 features that they can code. They respond to a size of line and a receptive field moving in a particular direction. All right. So here we see, end stopped, where this is now a horizontal line moving downward. It responds best if it's this size. If it's smaller than that, it responds less. If it's bigger than that, it responds less. If it moves in the opposite direction, if it's the right size but moves in the opposite direction, it doesn't respond. So 3 features that are coded. And how this happens, it's all due to divergence. To those donut center surround receptive fields sending their axons, if they belong to that particular thing, if they belong to that particular thing, that particular feature, to that particular hypercolumn. Good? We'll talk about how we get direction-specific signals when we talk about movement, and that'll be in the second section of this class. So first exam, we're just going to try and get through basically bottom-up signaling. Second section, we're going to talk about complex processing like color, movement, depth perception, selective attention, all of that stuff. Okay. All right. Right now, slide 41 and 42. 41 and 42. Summary of all the types of cells we've talked about and what they do and what their receptive fields are. This is a good table to memorize and understand. All right? So essentially we take this really complex analog signal coming into our brain, break it down into bits and pieces, action potentials, in center surround donut receptive fields in our retina that is already processed to enhance edges, send it to our LGN that gets filtered for attention, and then diverge it to recreate. We have to reconstruct from these bits and pieces what we're seeing, objects, colors. We have to reassemble it in those 250 million neurons. All right. So far we've talked about lines, angles of lines, right? Talked about light, dark areas. But this is not what we see in our world, right? We see complex shapes. And you notice that this is gray, sort of grayscale. I'm not even introducing color into the picture yet, because that adds a whole other layer of complexity to it. So if you're trying to detect this visual scene, what are the things that are important for you to detect here, if you want to interpret that? What are you trying to see here? Well, just describe to me, what do you see in this picture? Outline of the building, lines, what else? How are you able to tell the different buildings apart? What? You gotta speak loud on the floor.
So we can tell from shadows and also the difference in size.
Difference in size, difference in darkness, intensity. Like, how do we know that, is this a different building than this building? Yeah, because it has different intensities. What about this one over here? Does this belong to this building? Are these the same building? No? Why not?
Contrast.
The contrast, yes, the contrast, right? So we have these dark light patterns. We have areas of low spatial frequency, not a lot of change, and that can be light or dark. And then we have areas of high spatial frequency, where it almost looks like stripes. There's bricks, there's patterns. So changes in spatial frequency and contrast, is an important detection feature for us. And there's actually simple cells, there's V1 cells that respond to high contrast and low contrast. It's a primary feature that we detect. So high contrast means that there's very sharp edges. Low contrast, either no edges or the edges are more blurred. Here we can see high contrast and high frequency. Here we see low contrast, high frequency, low contrast, low frequency. What's important here, well, there's a lot of things that are important, but one of the things that's important here is, thank you, Marianne, is that you can see that with higher, well, probably from further back you can't, but with higher contrast and higher frequency, we have better detection capabilities. You can see these lines better. These with lower frequency, it becomes harder to see the edges. And as we decrease, even if we have good edges, as we decrease the contrast, it starts to fade away and we're able to no longer see that. It becomes grayish. Even though those lines exist, we're not able to detect them as well. So there's an interaction with our threshold for detecting things between contrast and frequency, spatial frequency. All right. Talk a little bit about adaptation. Hopefully you guys remember adaptation, sensory adaptation. If you have repeated stimuli over and over again, you become less tuned to that. It becomes harder to detect that. And that can happen at multiple different levels. It can happen at the level of the receptors, where neurons get fatigued. They get sort of worn out. If they're being constantly stimulated, that photopigment's getting used up, and you get a smaller and smaller signal out of those receptor cells, which means that the perception gets harder and harder. It can happen at the level of the LGN. Because that signal is coming in, it becomes sort of normalized. We start to tune it out. And it can happen at the level of the cortex. Sorry, filtering cortex kind of did that opposite. Shift in attention and what does stimulus salience mean? What does salience mean? How distinctive it is, exactly, if it pops out at you, if it's different, right? And then if something's constantly present, it becomes less and less different. All right. So we can actually use the fact that our brain does this thing called adaptation to sort of demonstrate that certain receptors detect certain things, because we can fatigue them. So when we talk about color, when we talk about trichromatic color theory, right? If I put a square of blue light up here and make you stare at it, and then all of a sudden I stimulate with equal light, you'll see an after effect of yellow, because when we stimulate with equal light, we've worn out the blue. So when we try and stimulate all the receptors, all we get are red and green light, which combines to be yellow. So you see that yellow. So we can use this to sort of understand through psychophysics, present a stimulus, ask you what your perception is, and by looking at sensory adaptation, we can start to understand what neurons are doing in our brain, which is pretty cool. Without sticking electrodes in your V1 like they did in the cats, right? We can actually see that, yes, humans do this too, because we can use psychophysics to understand that. All right, so I want you to stare right at this bar. Everybody's staring at the bar. Keep staring at the bar. We're going to do a lot of staring in here. Stare at the bar. And what do you see there? What happened when I switched the slide? What did the lines look like? They were shifted to the opposite direction, but now they're what? Vertical, right? So if you do this, what you're doing is you're causing adaptation. You're reducing the sensitivity for that column that's responding to that angle of line. It's being stimulated less and less and less in your V1. And then when I turn to this, you get a rebound effect where you see the opposite angle, and then as it recovers from being fatigued, it straightens up and becomes the actual stimulus. Pretty cool, right? You should do that on the PDFs that have one slide per page. Go, like, pull up this one and do that from a computer, and then go to the other one. It's really cool. You can like watch the lines bend back to normal. It's really cool. It's probably hard in here being further away from the screen. All right, so if we were to go into your V1, and here's the neuron that's going to respond to the second stimulus best, a vertical line. It's got a tuning curve for a vertical line. Well, what's happening when we keep presenting this line, the first stimulus? When we're keeping stimulating over here, we're wearing out this side. We're sort of fatiguing and wearing out this side. This side here is becoming less and less able to stimulate. So when we do, it's decreasing its activity. It's fatiguing. Is this area fatiguing? No, because we're stimulating with that angle of line. But then when we show the vertical line, and it should respond this way, it can't, because we fatigued this side. So when we first show that, the biggest stimulation is for the rebound, the other side. So we perceive that because it's the most activity. And then as it recovers, it shifts back to its normal tuning curve. Isn't that cool? No? You're like, what is it? I just think that's like, wow, we can understand these sensory illusions because of the way our V1 is built with these tuning curves and these orientation curves. All right, so stare right here at the center of this. Keep staring at it. As you stare at it, is your perception changing?
The edges are kind of like smoothing out.
Yeah, it's becoming sort of less able to detect it, right? It's almost like fading away. What do you see now?
It's like fading back.
It's like flickering in and out, right? So, like, shake your eyes off. Like, now look back, right? This is the same spatial frequency, but just lower intensity. We adapted to that, so it's almost like we've reduced that curve so much that we can't see, when we've adapted to that, this less intense signal. All right, do it one more time. Look at this one. We're adapting, we're adapting, we're adapting. What do you see now? When that first happened, so now we have a less intense horizontal frequency here. But when I first switched to it, you probably saw a checkerboard, right? Because you had adapted to this, you saw that after image superimposed on the new thing. And as your tuning curves recovered, your spatial this is how we know we have spatial frequency tuning curves, because we can produce adaptation to spatial frequencies. All right, so if we're looking at this image of this guy here, there are parts of this image that are high frequency, parts that are low frequency, parts that are in between, and this is how we reassemble in columns that respond to individual parts to changes in light-dark areas, and we mesh those together to reassemble into our final perception. right? Okay. I want to show you this. I'm running out of time. Basically, I'm going to skip forward in this video, but
more and more we will be able to measure and understand how our brain cells function.
they have,
We present this to the animal.
They're
and
presenting-
record the activity of the visual neurons. And these electrical signals that we recorded from will travel through these cables.
She managed to tap the vision from the brain of a cat and to reconstruct what the cat sees onto a computer screen.
So this is a digitized movie. This particular movie is a short clip from Indiana Jones, I think.
Here we see the two images next to each other. On the left, the image that was shown to the cat.
this is a
On the right,
receptive
the image
field,
that is tapped out of the cat's brain.
and that's what the neurons responding
The
to.
picture has a lot of random flickering, the noise.
Yeah.
That's probably the noise of the actual neurons. Because they sometimes fire spontaneous spikes. We consider those noise, but maybe they reflect something else, maybe.
All right, so we're actually able, like, I just took a screenshot of this. So here's the guy, the image of the head of the guy looking, and this is what they were able to record from the receptive field of V1, right? It's probably a low spatial frequency neuron. It's not a high spatial frequency because it's more blurry, right? But we're actually beginning to be able to map and like see like how our brain sees, like how it does this. It's pretty amazing. And we don't need a lot of spatial frequency in order to detect things. If I show you this picture, raise your hand when you know what this is. Almost immediately, what is it?
Abraham Lincoln.
Yeah, Abraham Lincoln. Very distorted, very pixelated, low spatial frequency. You don't need a lot of details. I mean, it could very well be some other guy with a beard, right? But anyway, we don't need a lot of information to be able to do this, right? All right. This is just sort of a summary of all this. All right, last thing. We're going to get out of here on time. It's amazing. Remember this that I showed you? Ocular dominance columns, left eye, right eye. When we're born, we don't have that. When we're born, our cortex looks smooth like that. There's no left eye, right eye input. Because before we're born, what type of vision do we have? What type of environment are we in before we're born? Dark, in uterine, right? We're prenatal, there's no vision. We have to have light. Experience is necessary in order for our visual system to develop. And so we see the appearance of these happening, and we get the ocular dominance columns appearing around week 5, and by week 13, they become sort of cemented in place. So this isn't all like genetically programmed somehow to like, poof, there it is in development. We have to have experience to encode these features, and that's going to be a common theme across the sensory systems is that experience is critical to development. And lack of experience, deprivation, not providing experience can have really bad effects. So for instance, if we take an animal and we raise them in an environment that only has vertical lines and they never see another orientation of line, guess what? They don't develop columns. They don't develop columns to detect other orientations of lines. You have to have the input from all various orientations of lines at different places in order to develop the ability of the brain to process that signal. And so a cat that's raised in a vertical world can only detect these lines and is blind to these angles. A cat raised in a horizontal world is blind to these angles. Cortically blind. They don't have the brain becomes cemented in place as it develops in these columns, and if we don't get that experience, then this happens. If you have astigmatism early on, a distortion of the image coming into the retina that prevents seeing a particular type of feature, this can happen in people, right? And so it's why in little kids, very early on, they're trying to make sure that vision is good, that there's no deficits, and if so, we're going to get glasses to correct for that so that we can make sure that everything develops properly. All right, we're out of here. That's a lot. You guys are troopers. Thanks for being here today. I'll see half of you for lab today. The rest of you I'll see at 8 on Thursday.